Thank you very much to give us opportunity to present our work in ISMB 2020. The topic of today's poster presentation is Novel in silico skin sensitization prediction model for cosmetic ingredients by analyzing their affinity towards CD54 and CD86 receptor. I'm Mr. Sunil Jayant from Annotation Analytics, the company located in Gurgaon, India. I completed this work with my colleague Sara Akuremi, and for any query you can contact on my email address sunil.jayant@annotationanalytics.com. So first question, what is skin sensitization? So skin sensitization is one of the signaling pathway where your external compounds like cosmetic ingredient, chemical compounds, or any external API interact with the skin, and it leads to sequence of pathways. So the first reaction is binding of compound on the skin protein, and this process is known as a haptenization. After binding of those compounds, there is an activation of cytokinin and then proliferation. This cytokinin leads to activation of Langerhans cells and then further activation of dendritic cells. And dendritic cells matured and then amplified and matured leads to formation of clonal expansion. This process known as a clonal expansion and leads to formation of T lymphocytes. So this sequence of pathway leads to allergic response on the skin. And when this cycle continues, so there is a more illicit reaction, so called like a skin sensitization. So here we are talking about, out of all those pathway, the key event 3. And the key event 3 experiment are known as the HCLAT. So what exactly HCLAT? HCLAT is a human cell line activation test, and this test is recommended in the OECD guideline number 442E. And it's adopted a cell-based assay that contribute to the assessment of a skin sensitization potential of skin care ingredient. This is, as I said, it's a key event 3 of skin sensitization, which is the adverse outcome pathway for a skin sensitization. And the process is like that: when the compounds are incubated with the dendritic cells, they lead to expression of two receptor proteins, CD54 and CD86. And the compound which leads to expression of these two protein receptors are known as a skin sensitizer. And the compound where these proteins are not expressed are known as a non-sensitizer. So positive compound sensitize, positive sensitizer means the compound which express these two proteins, and negative which are not expressing these two proteins. So here, what is our objective? Because the limitation for these kind of experiment, if you have a more number of compounds, it's very difficult to screen all the compound because this is very time consuming and it's not very cost effective. So in that case, so we are developing one in silico model, and we consider the compound which are a skin sensitizer, they have more affinity for CD54 and CD86, and the compound which are non-sensitizer have low affinity for CD54 and CD86. So in that case, we are exploring molecular docking approach. So in our method, first we obtain protein structures of CD54 and CD86 from RCSB protein data bank, which are here. Then we obtain the compounds which are mentioned in OECD guideline of HCLAT 442E. Then we perform molecular docking between compound and the protein, and then after molecular docking, we estimate the affinity. So if we have a higher docking score, means a strong affinity, the compound is sensitizer. And if we have weak affinity, means lower docking score, the compound is non-sensitizer. In our results, as we mentioned, so we took 4 skin sensitizer from OECD guideline, and which are expressing CD54 and CD86. And in our results also, we found strong affinity for those compounds with CD54 and CD86. We have also selected 4 compounds which are non-sensitizer, means they are not expressing CD54 and CD86, and also we found weaker affinities for those compounds through our docking approach. So in this table, we are summarizing all the results. So rank number 1, 2, 3, 4 are those compounds which are mentioned as a positive sensitizer in OECD guidelines. So these are the 4 compounds, and then where you can see the interaction with CD54 and CD86. So more docking score. and the 5, 6, 7, 8 ranking compound are non-sensitizer or negative sensitizer, and we are finding very low affinity for those compounds. So we separated two compounds into positive sensitizer with strong affinity and negative sensitizer with average to low affinity. We looked here at the diagrammatic representation of binding of these compounds on two different proteins. So here 2,4-dinitrochlorobenzene, which is very strong sensitizer, red mark showing positive sensitizer, are binding very strongly on the CD54 and CD86, and this is the docking score for 2,4-dinitrobenzene for 54 and 86. We selected representation for one compound which is negative, means non-sensitizer. So this is the binding of glycerol on CD54 and CD86. Here you can see the docking score, which is very lower than the previous strong sensitizer. So this is the typical binding. So here our conclusion is like that. So our computational results present the proof of concept of in silico method, where positive sensitizer making strong affinities with the receptor, then negative sensitizer compounds. In silico screening can be combined with in vitro method to reduce cost of pre-screening of compounds and get alert for sensitization potency of compound, which will be validated by later by in vitro experiment. For getting more details of this assay, you can go for OECD guidelines 442E and also the second reference to get more details. So thank you very much for your attention, and then I acknowledge Annotation Analytics for all the support for performing this work, and Sarah Akrami to support us. Thank you very much.